Welcome back. So, in today's video lecture, we will look at the demonstration of emulsification using various emulsifying devices. As part of demonstration, we will show you how to uh, perform an emulsion using an emulsifier. In classical, people use surfactant, as an emulsifier, okay, to perform an emulsion. But in modern days, nanoparticles are also being used. So if you choose nanoparticle or any colloidal system as an emulsifier to emulsify the mixtures, such process is called Pickering emulsion. Okay, and in today's example, couple of my students will demonstrate to you how to perform an emulsion using the nanoparticle. Basically, they will show you how to make an oil in water or water in oil emulsion using two major emulsifying devices like homogenizer and the probe type sonicator. Okay, let's begin.
Welcome back. My name is Vishal Singh Pawak. I am a PhD student in the Department of Chemical Engineering, working under the supervision of Dr. Maniganna sir. Today, I will demonstrate emulsification using several emulsifying devices. Initially, I will explain the working principle of two devices. One is the prototype sonicator that we have, and another one is the homogenizer. Then we will proceed with the demonstration part. Welcome back, everyone. In today's video lecture, we will explore two commonly used emulsifying devices, the prototype sonicator and the homogenizer. These devices are widely employed across various laboratory and pilot scale settings. They are particularly useful for applications like dispersing dry powders or nanoparticles into liquids, performing emulsification and stabilizing emulsions. For example, if you want to ensure that nanoparticles are evenly dispersed in a liquid medium or break up droplet aggregates and emulsions, these devices play a key role. Among the tools you might encounter, prototype sonicator and homogenizer are frequently used. Additionally, you might have seen ultrasonication baths, which are particularly popular for cleaning purposes. However, prototype sonicators are most commonly used for tasks like emulsification and nanoparticle dispersion. Today, I'll provide you with an overview of prototype sonicator and the homogenizer. We will then proceed to our demonstration part, where students will show you how to use these devices with specific samples. Let's start with the prototype sonicator. As the name suggests, this device produces sonic waves using a probe attachment. This probe is directly immersed in the sample and the sound waves generate from it, and it helps to agitate and mix the sample. You can, this is the closer look of the instrument. As you can see, this is the ultrasonic generator, basically helps to generate alternating currents. Another one is the transducer. It converts the alternating current into ultrasound. Another one is the booster horn. This amplifies the ultrasound energy. Another one is the sonotrade. It conducts the ultrasound waves into the sample. When the probe is placed into the sample, the intensity of sonication can be controlled by tuning the frequency, or we can say the amplitude supplied by the ultrasonic generator. If we talk about the key features, the energy is delivered into the sample in the form of sound waves, which cause agitations, aiding in better mixing. The ultrasonic bath frequency is about 20 kilohertz. Classify the devices ultrasonicator. Originally, such devices were deployed for cleaning purposes, especially for items like glass slides or sample containers. In nanotechnology, these devices are indispensable for dispersing nanoparticles into liquids. It can also break up aggregates of micron-sized particle, ensuring uniformity. In emulsions, probe-type sonicator are particularly effective by applying sound energy. They help break droplets during early stage of emulsification, creating smaller droplets compared to those produced by homogenizer. Advantage of producing smaller droplet size has been demonstrated through multiple research studies comparing the probe-type sonicator with homogenizer. If we will talk about the working principle of probe-type sonicator, the probe-type sonicator works through a process of compression and expansion. During this cycle, sound waves propagate through the sample. leading to cavitation, which is the formation of bubbles. These bubbles grow over time until they reach an unstable size. When the bubble collapse, they release a significant amount of energy. This energy is what creates intense agitation within the sample, ensuring effective mixing and particle dispersion. Now, next move to the another device, that is the homogenizer. The homogenizer is a device primarily used to achieve uniform mixing and emulsification in liquid samples. Its design and operational principles make it highly effective in various industrial and laboratory applications, especially when dealing with large sample size. Unlike the probe type sonicator, the homogenizer operates based on a rotor stator assembly, which plays a crucial role in its functionality. If we talk about how it actually works, it consists of basically stator and rotor. Rotor are a rotating element that generate high speed motion. If you talk about the stator, a stationary element surrounding the rotor with slots or perforations. Now, if we talk about how this device actually works, when the rotor spin at high speed, creating a suction effects that draw the liquid sample into the stator slots. This movement generates an axial flow, meaning the liquid flow along the axis of rotation. As the liquid passes through the narrow gaps between the rotor and stator, it experiences intense shear forces. Used in this is used in biological research to homogenize tissue samples or extract proteins from cells. And if we talk about the continuous mixing case, this process liquid is expelled from the stator slot and re-enters the cycle. This repeated action ensured through an uniform mixing of the sample. And this also this forces also break down the large particles in or droplets into the smaller ones, leading to uniform dispersion or emulsification. Now, if we talk about the key features of the homogenizer, the speed of the rotor can be controlled and typically ranges from 3000 to 25,000. In advanced models, speed may exceed this range, offering more precise control over the emulsification processes. Adaptability to sample size. One significant advantage of this homogenizer is its ability to handle larger volumes efficiently, unlike the probe type sonicator, where efficiency decreases with increasing sample size. The homogenizer maintains its effectiveness across a wide range of batch sizes. If we talk about the efficiency, the intense shear and turbulence generated during operation result in consistent and reproducible outcomes, making it ideal for applications requires uniform mixing. If we talk about the application part of probe type sonicator and the homogenizer, one is the degassing and deaeration. It can remove they can remove the trapped air or gas bubble from the liquid, ensuring smooth and air-free emulsions or dispersion. Second is the emulsification, widely used for stabilizing emulsions in formulations such as creams, sauces, or pharmaceutical preparations. Next is particle size reduction. Break down larger particles or drop it into smaller, uniform size for applications securing nanoscale dispersion. If we talk about the homogenization, sample homogenization, ensures uniform mixing of diverse components in a sample, making it suitable for food, cosmetic, and chemical applications. And now the biological applications, disrupting cells to extract proteins or DNA. We talk about the food processing, homogenizing sauce and other food products, chemical mixing and reactions, facilitates efficient mixing of reactants, often accelerating the reaction rate and ensuring uniform distribution of reactants. And another one is cosmetics and personal care product formulations. In this, devices are indispensable in the production of lotions, creams, and other cosmetic products, ensuring smooth textures and stable formulations. If we talk about the advantages and if we talk about the comparison of both the devices, now, and what is the advantage of homogenizer? One is the high efficiency, another one is scalability. Another one, if we talk about the uh, versatility, and if we compare it with probe type sonicator, homogenizer can handle large sample efficiently, whereas the performance of probe type sonicator decreases with increasing sample size. And if we talk about the droplet size, probe type sonicator generally produces smaller droplet due to intense ultrasonic energy, whereas homogenizer focuses on uniformity over fine droplet sizes. If we talk about the application ranges, homogenizer are better suited for applications involving large scale mixings, while probe type sonicator excels in nanoscale dispersion and breaking aggregates. Now let's move to the demonstration part. Now that we have all covered the theoretical aspects, we will move to the practical demonstration. Our colleagues will show you how to use these devices with specific samples. Through this, you will see firsthand how probe type sonicator and homogenizer function in real scenarios. Let's conclude this session here and proceed to the hands-on segments. Thank you.
Demonstration on probe sonicator. Ultrasonication, also known as sonication, is a high frequency device, powerful device that breaks large particle into smaller fragments and is also used to achieve uniformly shaped particle in a liquid sample. This probe sonicator has versatile application in the field of chemistry, biology, material science, and food processing. It has three components. First one is generator, the second one is transducer, and the third is probe. So initially what generator does is generator mm, generator converts electrical signal into electronic signal, and it also controls the transducer. Then the transducer converts electronic signals into mechanical vibration, high frequency mechanical vibration. Then the probe amplifies those ultrasonic vibrations into longitudinal vibration, creating a cavity in the sample. Here we have to put our sample holder. Here is the place for sample holder, which should be fit in size with the probe, and it should be made of glass or plastic, and it should be compatible with the sample. So this probe sonicator has utilizes high frequency ultrasonic sound waves, creating vacuum bubble in the solution. And during this process, during this kerridation process, these bubble form and collapse, releasing energy through shock waves into the system. And then this energy weakens molecular bonds, allowing particle in the solution to disperse and mix effectively. Now we'll switch on this equipment, probe sonicator. As you can see, it uses 750 watt and 20 kilohertz of the high frequency. As the display shows here, it shows temperature, time, temperature, pulse, and amplitude. Here is 4 and 5 function that is attached to the probe sonicator. Initially, timer, which shows the duration of the ultrasonication process. Here is the pulsar. It is usually used in biological sample, but it on and off the sonication process. Then here is the temperature. We can set probe temperature and temperature of the setup in ultrasonication process, but as here you can see, there is no digital thermometer attached to this equipment. So initially we do the experiment at room temperature. Then here is the amplitude. We can set this amplitude from 0 to 100%. This amplitude is the strength at which the ultrasonication happens. Now here the last one is energy. It is the amount of energy transferred into the system, which can be shown here in joules. Now we will shift to our system. This is a oil and water solution. We will set it to for probe sonication. We place this in our sample holder and we set this up for sonication. Now we'll switch on this equipment and wait for about 1 to 2 minutes. After this process has been complete, we can put a liquid sample in the microscopy and analyze the result uh, as the product can be seen as emulsion. Now we can shift to the application part of this probe sonicator. Through probe sonicator we can achieve uniformly sized particle in the last, and it also helps to disrupt DNA cells and to extract protein. Initially, what it can also be used for is cleaning and de-gassing. It can remove contaminants from the surface, and it also helps for evaporation of dissolved gases in the liquid sample. So we can conclude that this equipment can be used to for homogenization of the samples, and it is a very powerful tool for effective results. Thank you. Welcome to our demonstration on T25 Ultra Turex homogenizer. It is the switch on and off switch. Then this is the RPU from low to high, and this is the shaft. We can unplug the shaft and afterwards clean it from here. Now, this homogenizer can homogenize, disperse and mix samples. It has two parts, rotor and stator. The rotor part has high shear forces, and then the stator parts provides a stable platform for mixing. The speed through which it can move is from 3000 rpm to 25,000 rpm. It has a unique ability. Even with a small rotor diameter, it can achieve high circumferential speeds. The rotor stator design uh, is very effective in breaking down large particles, mixing substances, and creating stable emulsion. This homogenizer has various applications in the field of sonochemistry. It can also homogenize water sample, liquid sample, biological samples. It can help in particle size reduction and emulsification of the sample, degassing, and then cell destruction. Now we will see how the speed varies of this homogenizer in a water sample. So we need to note what kind of RPM requirement is there in different kind of samples. So here I have taken some amount of water in a beaker. Now I'll put this inside the sample holder. From here I'll detach the homogenizer and lower it in the water, and now I'll switch it on. As you can see, I have keep it in a low RPM. The RPM that you see here is RPM into 1000. As it is showing 3, so it is 3000 RPM. Okay. So now as you can see here, it has started homogenizing the sample. As we increase this speed, we can see that the homogenizing is happening at a faster RPM. Now we are at 6,000 RPM. This is at 10,000 RPM. So we see that we can go high as 25,000 RPM and low at 3,000 RPM. We have prepared the sample. We will run it at 10,000 RPM for 2 minutes, and we will then analyze the result in microscopy within trend with the probe sonicator and see what was the difference between the result. Now here I'm placing the sample at sample holder. Now we just put this in the sample. Here we see that the emulsion formation has been obtained, which we will be analyzing under microscope. Thank you.
Welcome everyone. We are here to analyze the oil and water emulsion formed using the different emulsifying devices. Here we have with us inverted microscopy that we are going to use. We analyze the droplet size of the emulsion, and here we have the emulsion. One is formed using the homogenizer and another one is formed using the probe tech sonicator. Here I have already prepared the slide and placed the sample on it. You can see here. Next, we will capture a series of images to compare the droplet size generated by both devices. Now, because of time constraint, we are not here able to show you all the images, so we will show you some few images. Here, let me show you one image. This is the images formed. These are the droplet size formed using the homogenizer. And another one we have is with POV type sonicator. So by visually examining these images, we can clearly see the difference between the droplet size from both the devices. These empirical evidence will provide valuable insights into the performance variance between the two devices. Thank you for joining us.